Today, my students, we will start with the pharmacodynamics. As we mentioned, pharmacology is divided into two parts: pharmacokinetics and pharmacodynamics. Uh, Pharmacodynamics describes the action of a drug on the body and the influence of a drug concentrations on the magnitude of the response. Most drugs exert their effect both beneficial and harmful by interacting with receptors, that is, specialized target macromolecules present on the cell surface or within the cell. The drug receptor complex initiates alteration in biochemical and or molecular activity of a cell by a process called signal transduction. Most of the drug targets receptors are protein molecules. All rules need exceptions, and many antimicrobial and anti-tumor drugs, as well as mutagenic and carcinogenic agents, interact directly with DNA rather than protein. Let's go now to the types of receptors. There are 4 types of receptors. Pharmacology defines receptor as any biologic molecule to which a drug binds and produce a measurable response. Thus, enzymes, nucleic acids, and structural proteins can act as a receptor for drug or endogenous agonists. We will come to discuss it within this course. However, the richest source or sources of therapeutically relevant pharmacologic receptors are proteins that transduce extracellular signals into intracellular responses. These receptors may be divided into 4 families. We will come also to discuss it through this course. Ligand-gated ion channel is the first. Gproteincoupled receptor, enzyme linked receptors, intracellular receptors. The type of receptors a ligand interact are depend on the chemical nature of the ligand. Hydrophilic ligand, which is water like, interact with receptors that are found on the cell surface, like in the figure 1, A, B, C. Please, when you hear this lecture, please look to the figures in order to make sure that you will understand what is the meaning of the type of the 4 type of or 4 families of the receptors. In contrast, hydrophobic, which is lipid-like ligand, enter cells through the lipid bilayer of the cell membrane to interact with receptors found inside cell. The figure 1D, which is included with with this in front of you presentation. These are 4 type: A, B, C, D. The 4 families or 4 types of receptors that is present in our body. please keep your attention with me to understand what are these receptors or types of receptors. Transmembrane ligand-gated ion channels, the extracellular protein ligand-gated ion channel usually contain the ligand binding site. This site regulates the shape of the pore through which ion can flow across cell membrane, as shown in figure 1a, which what will do regulates the entrance and the outflow of the ion from the cell to the cell, from the cell to the cell, inside the cell and outside the cells. The channels is usually closed until the receptors is activated by an agonist, which opens the channel briefly for a few milliseconds depending on the ion conducted through this channel. These receptors mediate diverse functions including neurotransmission and cardiac or muscle contraction. So it is mostly found in the neurons and the muscles in the body. So, for example, stimulation of the nicotinic receptor, by acetylcholine results in sodium influx and potassium outflux, generating an action potential. I think you take it in the physiology in the second class, in a neuron or concentration in skeletal muscle. On the other hand, agonists stimulate of the GABA aminobutyric acid receptors, increases chloride influx and hyperpolarization of a neuron. Voltage-gated ion channels may also possess ligand binding site that can regulate channel function. For example, local anesthetic that's used sometimes for the anesthesia of the gum that in the mouth, found in the mouth, bind to the voltage-gated sodium channel, inhibiting sodium influx and decreasing neuronal conduction and making the local anesthesia. The second type is the transmembrane G-protein-coupled receptors. Please, when you read or hear the lecture, look to the presentation, to the picture above. The extracellular domain of this receptor contains the ligand binding area, and the intracellular domain interacts when activated with a G protein or effector molecule. There are many kinds of G protein, for example, GS, GI, and G alpha, but they all are composed of 3 protein subunits. The alpha subunits bind guanosine triphosphate GTP, and the beta and GABA subunit enter the G protein in the cell membrane, figure 2. Binding of an agonist to the receptor increases GTP binding to the alpha subunit, causing dissociation of the alpha GTP complex from the beta and GABA complex. These two complexes can then interact with other cellular effectors. Please look to the picture again I concentrate on this. Usually an enzyme, a protein, or an ion channel that are responsible for the further actions within the cell. These responses usually last several seconds or minutes, so more than the first family in the action. Sometimes the activated effectors produce second messenger that further activate other effector in the cell, causing a signal cascade effect. So it is a scheduler, what's called effects. In the first, the receptor at the cell wall of is activated, then there is a series of effects until we reach to the effect that we want inside the cell. A common effector activated by GS and inhibited by GI is adenylyl cyclase, which produces the second messenger, cyclic adenosine monophosphate. G alpha activates phospholipase C, generating two other second messengers, inositol 1,4,5 triphosphate and diacylglycerol. Glycerol and cyclic AMP activate different proteins kinase within the cell, leading to a myride of physiological effects. Inositol phosphate 3 regulates intracellular free calcium concentration as well as some protein kinases. These we will come to discuss it more and more. So please take it as it is. What is mentioned in the front picture. Now we will come to the third family, enzyme linked receptor. This family of receptors consists of protein that may form dimers or multi-subunit complex. Please also look to the picture or the presentation to the right. It makes complexes. When activated, these receptors undergo conformational changes يتغير شكله, resulting in increasing cytosolic enzyme activity. Depending on their structure and function in the figure 3, this response lasts on the order of minute or hour, so it is more effect in the period than the first and the second family. The most common enzyme linked receptors, epidermal growth factor, examples, as an example, platelet derived growth factor, arterial natriuretic peptide, insulin, and others. Possess tyrosine kinase activity as part of their structure. This activate, please also look to the picture, all it is included, this explanation included in the picture, please, in order to understand what's going on through the activations of the receptor. So now we come, this response lasts on the order of minutes or 2 hours. The most common enzyme linked receptor, as we mentioned, possess tyrosine kinase activity as part of their structure. The activated receptor phosphorylates tyrosine residue on itself and then other specific protein. Phosphorylation can substantially modify the structure of the target protein, thereby acting as a molecular switch. So there is a many phosphorylation one by one. For example, when the peptide hormone insulin binds to two of its receptor subunits, their intrinsic tyrosine kinase activity causes autophosphorylation of the receptor itself. In turn, the phosphorylated receptor phosphorylates other peptides or proteins that subsequently activate other important cellular signals. This cascade of activation results in a multiplication of the initial signal, much like that with G protein coupled receptor, or looks like. The last fourth type, intracellular receptor. Now we come to the special type of receptors. This fourth family of receptor differs from the third above considerably from the other three, as we mentioned, in that the receptor is entirely intracellular, so it is inside the cell, and therefore the ligand must diffuse or enter into the cell to interact with the receptor, as in the figure 2.5, in order to move across the target cell membrane. The ligand must have sufficient lipid solubility. The primary target of this ligand receptor complex are transcription factor in the cell nucleus. Binding the ligand with the receptor generally activates the receptor via dissociation from the variety of binding proteins. This means that this receptor, it will liberated from the proteins that transform this ligand to the site where it will make its activity in the nuclei, as we mentioned. The activated ligand receptor complex then it translocates to the nucleus, as we mentioned, where it often dimerizes before binding to transcription factor that regulate gene expression. This activation or inactivation of these factors causes the transcription of DNA into RNA and translation of RNA into any array of proteins, and as a result this protein will give the needed effect in the site of action. The time course of activation and response of these receptors is on the order of hours. It may takes the effect hours or days. For example, steroid hormones exert their actions on target cells via intracellular receptor. Other targets of intracellular ligands are structural proteins, enzymes, RNA, and ribosomes. For example, tubulin is the target of anti-neoplastic agents such as paclitaxel, which is one of anti-cancer medicines or drugs. The enzyme dihydrofolate reductase is the target of antimicrobial such as trimethoprim, and the 50S subunit of the bacterial ribosome is the target of macrolides antibiotics such as erythromycin, which we will come to discuss it within the antibiotics or the antimicrobials in the very near future. Thank you very much. Our lecture is in. Thank you for my students.